So the students, please on your video. I'll take the attendance. Okay. And then afterwards you can off it again. But for the time being, you on it. Respond to your roll calls. Roll number 1, present. Or roll number 2. Roll number 2 is present. Okay. Roll number 3,
Present, ma'am.
4, 5, 6,
Present, ma'am.
7, 8. Roll number 8, 9,
Present, ma'am.
10.
Present, ma'am.
Okay, you can off your video. Your attendance will be sent later. Okay. Okay, so students, you can see this screen. I cannot take your attendance because of the signal problem. Yeah, so I may ask any of the student, you please respond. If you don't respond, maybe I'll think that you are not attending the class. So Vivek, he's present.
Yes, ma'am.
Last class I was calling you,
Yes, ma'am.
and then you were not responding. I don't know whether you attended the class or not. Did you
I
attend?
was attending, ma'am.
But I called your name so
Yes,
many
ma'am.
times, but you didn't respond. No?
Yes, ma'am.
Please attend the classes regularly, because if you read these topics by yourself, it will take time to understand the thing. Okay, when you attend the class, at least you can grasp something and you read the same chapter from the textbook, then it becomes easier, isn't it? Okay, so we'll continue. We'll continue this protein structure. So continuing the protein structure. Okay, in the last class, I have mentioned that structural organization of protein, we can divide it into 4, isn't it? So number 1 is the primary structure. Number 2 is the secondary structure. Third is tertiary and quaternary structure. So how will you define, Vivek Bolo, how will you define primary structure?
Distinguishing between primary structure, secondary structure, and tertiary structure.
Yeah, how will you define, if somebody asks you, define primary structure, how will you define?
Primary structure means a straight polypeptide chain.
Straight polypeptide chain. That is primary structure. Do you define primary structure as straight polypeptide chain? You have studied the same thing in class 11 and 12 also, isn't it? Structural organization of protein. The same thing this is being repeated in your first MBBS. So roll number 1.
Yes, ma'am.
Yeah, what is your name?
Aditya Gupta, ma'am.
Yeah, Aditya. Bolo, Aditya, how will you define primary structure?
Ma'am, primary structure is defined as a sequence of amino acid
Sequence of amino acids.
in linear fashion.
Sequence of amino acid which are connected by a peptide bond.
Peptide bond. Yes, ma'am.
Isn't it? So in the primary structure, what is important is the sequence of amino acid. Isn't it? Then secondary structure, how will you define?
Secondary structures are of 2 types: alpha helix and beta pleated sheet.
Okay. So here the amino acids which are far away from each other or which are near to each other are connected. Amino acids which are far away from each other or which are nearer to each other are connected in the secondary structure.
Near.
Yeah. So in the secondary structure, the amino acids which are near to each other are connected, to form typically 2 type of secondary structure. One is alpha helix and another is beta pleated sheet. Okay? And roll number 2, how will you define tertiary structure? Roll number 2 is present or not?
2 is absent, ma'am.
2 is absent. No, okay. Okay. Then 3.
Yes, ma'am.
Yeah, what's your name?
Akima Disangma.
Akima. Okay, Akima, tell me, how will you define tertiary structure?
Ma'am, it's a 3-dimensional arrangement of protein structure.
Okay. And here the linkage is between the amino acids which are near or far away from each other. What do you think? What do you think?
Ma'am, it's far away from each other.
Yeah. So amino acids which are far away from each other, they are linked together to form a 3-dimensional structure, which is known as the tertiary structure. And roll number 4.
Yes, ma'am.
What is your name?
Ma'am, Arkit Bhattacharya.
Okay, Arkit. Arkit, bolo, quaternary structure, how do you define?
Ma'am, it is the aggregation of certain polypeptides.
Polypeptides. Okay. If I ask you myoglobin, whether it has got a tertiary structure or not, myoglobin molecule,
Myoglobin, yes,
it will have a quaternary structure.
yes, ma'am.
Do you think myoglobin? How many polypeptide chain it has got? Myoglobin. ठीक है. Roll number 5.
Yes, ma'am.
Yeah. What is your name?
Aldenberg Khashandi.
So Aldenberg, you tell me whether myoglobin will have a quaternary structure or not. Myoglobin, where is it present?
Globin is present in the muscles.
Yes, in the muscles. What is the function of myoglobin? What do you think? What it does?
Storage of oxygen.
Oxygen, isn't it? So when you are doing any muscular exercise, no, just to continue the glycolysis, oxygen has to be supplied in the muscle. Okay, so that oxygen is utilized during muscular exercise. So how many chains it has got? Because it is also a protein, no? So how many polypeptide chain it has got? Any idea? ठीक है, myoglobin you forget. Hemoglobin, how many polypeptide chain it has got?
2 alpha, 2 beta.
2 alpha and 2 beta. So whether it has got a quaternary structure? ठीक है, Anup बोलो.
Yeah, ma'am, hemoglobin or myoglobin?
Hemoglobin will have a quaternary structure.
Yes, ma'am.
And what about myoglobin?
No, ma'am, only one polypeptide chain.
Okay.
Tertiary structure.
Yes, good. So myoglobin has got only one polypeptide chain. So the level of organization it can go is only up to tertiary structure, isn't it? So please listen, students, carefully. To call any protein will have a quaternary structure when they have got more than two polypeptide chain, two or more than two. Okay. A protein that is having only one polypeptide chain cannot form a quaternary structure. No, this is a basic concept. So a protein will have a quaternary structure when it has got two or more than two polypeptide chain. Okay, myoglobin has got only a single polypeptide chain. So it can have a primary, secondary and tertiary structure, but not a quaternary structure. Clear? So the two types of secondary structure that you have studied is alpha helix and beta pleated sheet. So this is alpha helical structure, isn't it? So it is in form of right-handed helix, and there are so many characteristics of alpha helix which you can go through again from the textbook. Then these are beta pleated sheet. You can see the orientation. These arrows are pointing towards the opposite side. So what type of beta pleated sheet it is from the structure? Can you make out? Vaishnav, bolo. Vaishnav, what type of secondary structure is this one?
Ma'am, I cannot see the screen.
You cannot see the screen.
No, ma'am.
Any other students who cannot see the screen? Okay, Jessica, because I cannot remember everybody's name, no? So whomever I know by name, I'm calling them, okay? So Jessica, can you tell me this type of structure, what type of structure is it? Secondary structure. Jessica, Anup बोलो ये वाला कौन सा type of structure है?
Anti-parallel.
Yes, anti-parallel, good. It is anti-parallel beta pleated sheet, isn't it? So if somebody asks you, this is a protein, what type of structures are present in this protein? You will say that it has got a secondary structure, that is the alpha helix, and two beta pleated sheets, no? Isn't it? Since the orientation of these two beta pleated sheet is on the opposite side, that's why we call it as anti-parallel beta pleated sheets. Okay. So here the carboxyl end is opposite. The two carboxyl ends will be opposite. So it is a anti-parallel beta pleated sheet. Clear? Now these two anti-parallel beta pleated sheet, they are connected by another type of secondary structure, which is known as the beta loops. So secondary structure may be either a beta pleated sheet, which may be parallel or anti-parallel, depending on the orientation of the ends, or it may be an alpha helix. Then the third type of secondary structure is loops, which is known as beta loops. Clear? Now this beta, what is the peculiarity of these beta loops? These beta loops are made up of mostly polar amino acids. So they tend to locate on the surface of the protein. Okay. Since they are made up of polar amino acids, they tend to locate on the surface of the protein. And one of the example is the beta loop, which is present in the antibody molecule. And this beta loop can act as a binding site for the antigen during antigen-antibody interaction. Clear? So this is an example of secondary structure, that is beta loop, mostly made up of the polar amino acid and located on the surface of the protein, and they tend to connect the two adjacent antiparallel beta pleated sheet. Okay? Any confusion? This beta loop. Then this is another type of secondary structure, which is known as the beta turn. Okay, beta turn is also made up of it also try to connect the two antiparallel beta sheets, or it tries to connect the beta sheet with alpha helix. So they are usually made up of proline and glycine. Okay, remember proline is the only amino acid which can form a cis type of peptide bond. So peptide bonds are usually in trans conformation. Okay, but if it is in the proline is the only amino acid which can form the cis type of peptide bond. So it can produce 180-degree bend. Okay, if proline is present, it can form 180-degree bend in a polypeptide chain. So proline and glycine is the smallest amino acid, so it can it can locate itself anywhere inside the polypeptide chain. Okay, so beta turns are usually made up of proline and glycine, and they produce the 180-degree turn, and it tries to interconnect the two antiparallel beta pleated sheet, or it tries to connect the beta pleated sheet with the alpha helix. Clear? So you have studied 4 types of secondary structure. One is alpha helix, beta pleated sheet, which may be parallel or antiparallel. Then number 3 is the beta loops, and number 4 is the beta turn. Beta loops, remember, it is like a hairpin loop structure. Okay, beta loops are hairpin loop structure, and they are usually present in a normal functional protein. Then in the last class, I asked collagen is having what type of structure? Anyone who could answer? Who can answer what type of structure collagen has got? Vivek, bolo, collagen, what type of structure it has got?
Tertiary, ma'am.
No, collagen ka structure, it will have a tertiary structure, but what type of secondary structure it has got? It has got the alpha helical structure, no? But there are 3 polypeptide chains which are having an alpha helical structure which winds on itself to form a typical triple helical structure. Okay, so remember in collagen, the structure is known as a triple helical structure because it has got 3 alpha helical chains which winds on itself. Okay, and remember collagen structure will have this particular amino acid sequence. There's 3 amino acid sequence: glycine, X, and Y. Glycine, okay, why is it present in collagen structure? Because this is the amino acid which is having a very simple structure and it can fit anywhere. Okay, then X represents proline, or it may be hydroxyproline, and Y can be any other amino acids. Okay, so the sequence goes like that: glycine XY, glycine XY. So glycine is present in every 3rd position. Okay, and X represents either proline or hydroxyproline. Okay, so collagen has got the amino acids hydroxyproline and hydroxylysine. I have already mentioned these are the amino acid which does not have any genetic code or doesn't have any transfer RNA to be inserted into the protein structure. They arise by post-translational modification of either proline or lysine. Okay, proline, when it undergoes hydroxylation, it will form hydroxyproline, and lysine, when it undergoes hydroxylation, it will form hydroxylysine. Okay, so this hydroxyl group is very important for cross-linking these alpha helical structures. Clear? So collagen is having what type of structure? It has got a triple helical structure. Clear? And the amino acid sequence which is present in the collagen structure is glycine, X, and Y. X may represent proline or hydroxyproline, and Y can be any amino acid. Clear? Okay. We will go to the super secondary structure. Okay. So this secondary structures like helix, beta pleated sheet, turns, or loops, they turn to associate itself to form certain motifs which are known as the secondary structure. Okay, this super secondary structure. Now the super secondary structure is mainly present in the domains of the protein. One protein may have many domains, and each domain will have different functional activity. Okay, that's why there is a term known as multi-enzyme complex. So multi-enzyme complex is nothing but an enzyme having different domains, and each domain will have different enzyme activity, but they are usually present in the single protein. Clear? So these secondary structures, what they do, they tend to associate to form these super secondary motifs. Example is beta, alpha, beta loop. Okay, here what happens is that this beta strand, it is connected to the alpha helix, which is again connected to the beta loop, okay, to the beta sheet by a beta loop. Okay, then this is known as alpha turn alpha. So there is an alpha helical structure, then beta turn, and again the alpha helical structure. This is another form of super secondary structure. So in the exam, they used to ask a short note on super secondary structure. First of all, you define what is a super secondary structure. Okay, give examples and you can draw some diagrams of super secondary structure and what is the functional importance of this structure. Clear? So one of the example is beta, alpha, beta loop, then alpha turn alpha, then it may be a typical beta motifs, okay, twisted beta sheets, or it may be a beta barrel. So there are different types of secondary structure. You can just show the diagram of around two or three. And then what is the importance? They are usually present in the domains of the protein and they have got different functions. Okay, now the super secondary structure may further associate with other super secondary structures to form the three-dimensional structure of the protein. Clear? Any confusion regarding the super secondary structure? Anup?
No, ma'am.
Okay, so what is super secondary structure? Can you just define it again? How it is formed?
Ma'am, it's by association of different types of loops, like beta loop
Association of different
and
types.
alpha chain again, then beta loop,
Yeah.
then alpha,
It may be an association of alpha
then
helix.
alpha turn alpha.
Yes, different types of secondary structure, no, like helix, beta pleated sheets, loops, and turns. They associate together
Yes, ma'am.
to form a super secondary structure. Okay, and you can give examples of few of this super
Yes, ma'am.
secondary structure and what is the functional importance. Okay, ठीक है.
Yes,
Now let us come to the physical properties of proteins. Okay, proteins, you know that it is crystalline in nature and they turn to form a colloidal structure in colloidal particles in aqueous solution. Okay, you know the oncotic pressure, isn't it? What do you mean by oncotic pressure? Anyone? Physiology also you have learned?
The pressure exerted by solutes.
Okay, so in the blood, no, this oncotic pressure is maintained by which protein?
Albumin.
Albumin.
Yes, good. Albumin. Okay. So albumin maintains the oncotic pressure inside the blood vessels, due to which the water is kept inside the blood vessels, isn't it? So whenever there is any deficiency of albumin, for example, if there is a burn, okay, and if there is a liver injury, for example, in liver failure, the manufacture albumin is synthesized less. So whenever the concentration of the albumin inside the blood decreases, what will happen? The water will migrate from the intravascular compartment to the extravascular compartment, producing a condition known as, leading to a condition known as edema, isn't it? So edema is the collection of fluid outside the blood vessels, no? Clear? So the importance of these proteins is that they are colloidal in nature in the aqueous solution and they maintain the plasma oncotic pressure. Then different proteins will have different molecular weight. For example, insulin has got 5,700 Dalton. Okay, and here in this chart, immunoglobulin is having the highest molecular weight. Now, just see the difference between the hemoglobin and albumin. The molecular weight of hemoglobin is less when compared to albumin. Okay, so whenever there is any injury to the glomerular membrane, no, whenever there is any injury to the glomerular membrane, what will happen? The albumin will come out along with hemoglobin. So when hemoglobin comes out into the urine, that condition is known as hemoglobinuria. Okay, remember, there are two terms known as hemoglobinuria and another is hematuria. So hemoglobinuria is the excretion of hemoglobin inside in the urine. Okay, and hematuria is the presence of RBC in the urine. Clear? So hemoglobinuria will occur whenever there is an injury to the glomerular membrane and hemoglobin cannot pass out from the glomerular membrane. So the lesion has to be within the renal tubules, isn't it? So whenever there is a stone in the renal tubule or injury to the renal tubule, only then RBC will come out in the urine. Clear? Any confusion? So remember, whenever there is any injury to the glomerular membrane, which occurs in glomerulonephritis and other conditions, the albumin will be excreted along with hemoglobin because the molecular weight of hemoglobin is less when compared to albumin. So in the last class, I have mentioned that the shape of the proteins are different. Some are globular, okay, like insulin, oval and globular, almost same. The polar amino acids in globular or oval proteins are present on the surface. That's why they are more soluble when compared to elongated or the fibrous protein. So in the Viva, if they ask you, give example of elongated or fibrous protein, you can mention fibrinogen, isn't it? Any other fibrous protein you know? Collagen, elastin, isn't it? These are all fibrous protein. Now isoelectric pH. So you know what is isoelectric pH, isn't it? So it is the pH at which the molecule will exist as Zwitter ion. So Zwitter ion means a molecule will have equal number of positive and negative charge. So the net charge will be 0. So when this type of molecules which are chargeless, when they are kept in the electrical field, they will not migrate towards the electrode. Yes or no? Okay. So proteins, each and every protein, there are different types of protein, isn't it, inside our body. So each and every protein will have its own isoelectric pH. So at that isoelectric pH, it will not migrate towards the opposite electrode. But on the acidic side, proteins will exist as, please tell me, on the acidic side, proteins will exist as, what do you think? Proteins will exist as, roll number 1, Aditya, tell me.
Ma'am, negative.
On the acidic side, what do you mean by acid? When you are adding an acid, that means you are adding a positive ion or a negative ion? Acid,
Ma'am,
hydro.
cation.
Yeah, you are adding a cation, isn't it? You are adding a proton. Hydrochloric acid
Yes, ma'am.
means you are adding a proton. So when you are adding a proton to a protein solution, it will acquire the positive charge, which is known as cation, isn't it? And when you add an alkali or the base, it has got the negative charge, so the proteins will exist as anion. Now this, so remember on the acidic side, protein exists as cation, and on the alkaline side of the isoelectric pH, the protein will exist as anions. Now these cations will migrate towards which electrode? Which electrode? Cations, roll number 1, you please tell me. Cations will move towards which electrode? Name the electrode. Aditya bolo. Cations move towards?
Ano?
Cations move towards which electrode? Jessica? Yeah. No, Jessica को बोलने दो. Jessica, tell me cations will move towards which electrode? It will move towards cathode or it will move towards anode? Just tell me. Cations will move towards cathode or anode. This is the basic things which you learned in 11 and 12. What is the positively charged electrode known as? Vivek बोलो. Positively charged electrode को क्या बोलते हैं?
Cathode.
Ma'am, cation will move to cathode and anion will move to
Cation.
anode.
Yes, that is the simple concept, isn't it? CCAA, remember. Cations will move towards cathode, which is the negatively charged electrode, and ions, which are the negatively charged ion, will move towards anode, which is the positively charged electrode. So there should not be any confusion, okay? And ions move towards anode, cations move towards cathode. So the proteins have got the amphoteric nature. Okay, so if they ask you what are ampholites or amphoteric nature of protein, protein on the acidic side can accept a proton and can act as a cation, and on the alkaline side, it will act as an anion. So it can act both as cation as well as anion depending on the pH. That's why they are known as ampholites. Clear? So these are examples of proteins which are having a different isoelectric pH. You can see pepsin is having an isoelectric pH of 1.1. So it is the protein which it is an enzyme or the protein which acts on the acidic side, isn't it? The biological activity of pepsin is maximum towards the acidic side. So since its isoelectric pH is also very low, 1.1. On the other hand, hemoglobin will have the isoelectric pH of 7.2, and likewise, so many other proteins will have different types of different isoelectric pH. Now let us see precipitation reaction. What do you mean by precipitation reaction? So precipitation reaction, okay, it can occur due to various agents. Number 1 is salting out. Okay, what do you mean by, so I will ask one of the students, please listen carefully. What do you mean by salting out or salting in? So salting out happens when you add a large quantity of a neutral salt like ammonium sulfate or sodium sulfate. So when you add this ammonium sulfate or sodium sulfate, what they try to do is they try to remove the hydration layer which surrounds each and every protein molecule. So remember, in aqueous solution, protein molecules will be surrounded by a layer of hydration because of the different types of charges that are present on the protein molecule. For example, the serine amino acid has got the hydroxyl group, isn't it? There is a carboxyl group, there is an amino group which tends to form a hydrogen bond with water. So there will be lots of water molecule around each and every protein. So it will form a hydration, which is known as a hydration layer. Okay, so each protein molecules, they are separated from each other by a hydration layer, thereby they are kept in solution. But once you add huge quantities of these neutral salts, what they do is they try to remove this hydration layer around the protein molecule. So due to which what will happen, the protein molecules will come closer to each other, they will try to interact with each other and they get precipitated out. This is known as salting out. Okay, so salting out occurs when you add a huge amount of ammonium sulfate, and this this ammonium sulfate, what they do is they remove the layer of hydration surrounding each and every protein molecule. Okay, so there is a saturation test. You can remove proteins by salting out method. For example, albumin is having a lower molecular weight when compared to globulin. Yes or no? Okay, albumin is having a lower molecular weight when compared to globulin. So albumin will have a more surface area when compared to globulin. So if the surface area is more, the layer of hydration will be more or less? Vivek, bolo. It will be more, isn't it? You have
Less,
got
ma'am.
कैसा less होगा? The surface area is more.
Ma'am,
A protein is having a bigger surface area, so there will be many amino acids, isn't it, on the polar amino acid which are present on the surface. So they tend to form a more hydrogen bond with the water. So the layer of water will be more. No? If the surface area is more. Yes or no?
yes,
So,
ma'am.
so if the protein is having a low molecular weight, it has got a bigger surface area, okay, and so a larger layer of hydration. On the other hand, globulin molecular weight is more when compared to albumin. The surface area is less, so the hydration layer is less. So it is easier to precipitate out globulin when compared to albumin. Okay, so half saturated solution of ammonium sulfate will precipitate globulin, but to precipitate albumin, you need a fully saturated ammonium sulfate solution. Clear? Any confusion? Anup, bolo, confusion?
Yep, ma'am.
So the bigger the surface area, there will be more layer of hydration. It is difficult to remove or precipitate out those type of proteins. Okay, so this neutral salt solution, what they do is they remove the layer of hydration surrounding the protein molecules, due to which the protein molecules tend to aggregate and then get precipitated out. This is known as salting out. Clear? Jessica, you please listen. I will ask you the same thing again. Salting out. Then there is another thing known as salting in. Salting in happens when you add a very small quantity of neutral salts. So when you add a small quantities of neutral salts, what they do is they try to neutralize those like charges. Okay, protein gets attracted to each other. Okay, when there is unlike charges, isn't it? So what they do is they try to neutralize those unlike charges which are present on the surface of the protein. Okay, so that the protein they repel each other because of more of like charges. So what will happen if they repel each other? Their layer of hydration surrounding each and every protein molecule will be more. Okay, so that is known as salting in. So salting in occurs when you add a small amounts of the neutral salts which try to neutralize the unlike charges which are present around each protein molecule, so that the protein molecules get repelled to each other. Okay, and there will be more layer of hydration in between the protein molecule. So salting in is to increase the solubility of the protein, and salting out is to precipitate the protein. Clear? ठीक है, Vivek?
Yes, ma'am.
So what is the importance of salting in?
Salting in is to increase the solubility of protein.
Solubility of protein. How does it increase the solubility?
When small amount of salt is added, the charges which are present in the protein disappears. So the hydration energy increase and the protein will solve more.
Yeah, for example, a protein, there are just imagine that there are two protein molecules, okay? One of the protein molecule will have a positive and both positive and negative charges, and another also positive and negative charges. So what this neutral salts will do, they will try to neutralize the unlike charges, no? In one protein molecule, it will neutralize the negative charge, okay, in both the protein molecules, so that all the protein molecules will tend to have a similar kind of charge, either the positive or the negative charge. So if they have all similar kind of charges, they will try to repel each other, isn't it? Okay, so that is known
Yes,
as
ma'am.
salting in. On the other hand, salting out, when you add a huge amount of this neutral salt, what they do is they remove that hydration layer around the protein molecule, due to which the protein molecule aggregate itself and then it gets precipitated out. Okay, so this is precipitation reaction. So precipitation reaction can be due to salting out. Then number second is isoelectric precipitation, which I've already told. Each and every molecule at its isoelectric pH will have no charge, so they tend to aggregate and then they get precipitated out. So can you anybody explain me how the curdling of the milk occurs? Anup, have you studied how the curd is formed?
No, ma'am.
So curd is because formation is occurs because of which bacteria when you store the milk?
Lactobacillus, ma'am.
Yeah, lactobacillus. So what this lactobacillus they do is that they cause the fermentation of the milk. No, they reduce the pH. Okay, so they bring down the pH to around 4.6. That is the isoelectric pH of the casein. Okay, so when the pH of the milk is reduced to 4.6, the casein becomes precipitated out, due to which the curdling occurs. Clear? So lactobacillus that is present in the milk, they tend to decrease the pH of the milk to around 4.6, which is the isoelectric pH of the casein, which is the milk protein present. And at this pH, the casein will get precipitated out, okay? And it will form a curd. Clear? So this is the precipitation at the isoelectric pH. Number third is the organic solvents. Organic solvents, what they do is they also reduce the dielectric constant. Organic solvents like ether, alcohol, chloroform. So what they do is they try to reduce the dielectric constant and remove the hydration layer surrounding the protein molecule. Okay, due to which the protein aggregates and gets precipitated out. Then again, proteins can be precipitated by heavy metal ions. Okay, it can be precipitated by alkaline solution, or it can be precipitated by acidic solution. For example, in the estimation of creatinine, have you done estimation of creatinine in your practical? Practical estimation of creatinine, it is completed or not?
Yes, ma'am.
Yeah, so what do you add in the estimation? Do you add some acid, yellow color acid, which is having a yellow color? Yeah, you add a picric acid, isn't it? You add picric acid, and what happens? Picric acid you add because proteins can be precipitated out on the acidic side or on the alkaline side. You have to remove the protein so that the protein does not interfere with the estimation of creatinine. So in any estimation, whether it is a urea estimation, whether it's an estimation of sugar, okay, estimation of glucose or estimation of creatinine, the first step is to remove the protein. Okay, and that protein is removed either by adding an alkaline solution or by adding an acidic solution. For example, in case of estimation of creatine, you are adding the acidic solution, that is picric acid. Yeah. So what is the reason for adding the picric acid is you want to remove the protein so that it does not interfere with the estimation of creatine or creatinine. Yeah. So precipitation can be done by various agents. It can be done either by salting out, it can be done at isoelectric pH, it can be done by adding organic solvents or acidic or alkaline solution. Okay, precipitations can also be done by forming an antigen antibody complex. So those things, so these are different methods by which precipitation can be done. So this is usually done in ELISA techniques or in the immunoturbidometric method. We are adding an antigen antibody complex and try to remove the proteins. So they are used in the estimation of creatine kinase or the isoenzymes of creatine kinase. Now let us come to denaturation. What do you mean by denaturation? So remember, in denaturation, there is a loss of secondary, tertiary, and quaternary structure. but the primary structure is preserved. Okay, so denaturation. In denaturation of the protein, there is loss of secondary, tertiary, and quaternary structure, but the primary structure of the protein is preserved. So what will happen to the biological activity of the protein in denaturation? It will be lost or it will still be maintained. For example, if
Lost.
yes, hemoglobin, if hemoglobin is denatured, okay, all its tertiary structures will be broken. That means the subunits will get dissociated from each other, and then there will be loss of tertiary and the secondary structure of each subunit. Only the primary structure will be maintained, but the biological activity of the hemoglobin to carry and deliver the oxygen to the tissues will be lost, because they will not exist together as a complete functional protein, isn't it? Because in hemoglobin molecule you have got a heme group, and in the center there is an iron which connects the different porphyrin. There is a center, iron is present in the center of the heme, isn't it? So it connects the different porphyrin groups. So if the all the structures is lost except the primary structure, the function of the hemoglobin will be lost. So denaturation can also occur because of heat, because of the agents like urea, vigorous shaking, okay, high pressure, UV rays. These are the agents which causes denaturation. So remember, in denaturation, the primary structure is not altered, but secondary, tertiary, and quaternary structure, everything will be altered. As a result, there will be loss of biological activity, and the protein can be easily precipitated out. So have you done heat coagulation test in practical?
Yes, ma'am.
The heat coagulation test, no. So a heat coagulation test, what do? You fill up the 3/4 of the test tube with a urine sample, okay, or with the protein sample, and you heat only the upper 1/3. And then after some time, what you will see? You will see a white precipitate, yes or no? You will see a white precipitate, and ultimately this, if you go on heating, the protein will form a coagulum, isn't it? So in coagulum, remember, it is an irreversible denaturation. Coagulum is an example of irreversible denaturation. So denaturation can be of two types, reversible and irreversible. So in the reversible denaturation, if you remove these agents, for example, if you stop heating, what will happen? The protein will get renatured again. But usually heat denaturation usually leads to irreversible denaturation. So if you remove these denaturing agents like urea, salicylates, the protein may revert back to renatured protein. Okay, but in case of irreversible denaturation, it cannot form a functional protein once the denaturation occurs. Example is coagulation. So all the coagulated protein, for example, when you boil the egg, the protein becomes coagulated, isn't it? So if you try to dissolve the coagulated egg in an aqueous solution, it will not get dissolved. Clear? So coagulated proteins are it is an example of irreversible denaturation. Okay, so this is a normal functional protein. When it gets denatured, all the structures are lost except the primary structure. And when the denaturing agent is removed, again, it will be converted back to its original structure. So I've already told it can be of two types, reversible denaturation and irreversible denaturation. And in the heat coagulation test, so you remember in the heat coagulation test, you are adding something, glacial acetic acid, yes or no?
Yes, ma'am.
So you add the glacial acetic acid because you want to bring down the pH of the albumin to around the isoelectric point, okay, or the isoelectric pH. So you heat the protein solution or the urine solution, you see some precipitate, then you add the glacial acetic acid, bring down the pH of the urine to the isoelectric pH of albumin, and then the albumin will get coagulated, or the precipitation will become more prominent. Okay, but if the phosphorus is present, what will happen? It will disappear. So there is one protein that is present in the urine which can be diagnosed by heat coagulation test. Any important protein which can be diagnosed by heat coagulation test, not diagnosed, which can be visualized by heat coagulation test, which is present in multiple myeloma. Do you remember any protein? So in multiple myeloma, that is nothing but the tumor of the light chains of immunoglobulin. Okay, it is also known as plasma cell tumor. So you get one typical protein, okay, which we can see in heat coagulation test, that is known as Benz-Jones protein. Remember or not, Benz-Jones protein? Do you remember? Aditya, do you remember Benz-Jones protein?
Yes, ma'am.
Yes, ma'am.
So Benz-Jones protein, if you want to see whether it is present or not, we do heat coagulation test. Okay, it gets precipitated out at a lower temperature when compared to albumin. Okay?
Yes, ma'am.
Then there are various chemical properties of protein. One particular test to identify protein is the Biuret test. Have you done Biuret test in the practical?
Yes, ma'am.
So any idea what is the principle of Biuret test? Biuret test, what is Biuret? If somebody asks you what is Biuret, what is Biuret? It is a compound when you add when you heat 2 molecules of urea. Okay, so biuret is a compound. When you heat 2 molecules of urea, it will form a compound which is known as biuret. So for any compound or any molecule to give the biuret test positive, there has to be at least 2 peptide bond. Okay, 2 peptide bond. So do you think an amino acid or a dipeptide can give biuret test positive? Amino acid, how many peptide bond it has got? हाँ आदित्य बोलो, amino acid में कितना peptide bond है?
Ma'am, no peptide bond in amino acid.
Okay, good. Dipeptide में how many peptide bonds?
1.
1. So remember, those molecule which does not have a peptide bond, which is having a single peptide bond, cannot give biuret test positive. It has to be at least 2 peptide bonds. Clear? So not only protein, but also compounds like urea, when it is heated, it can form the biuret. It can give biuret test positive. So the principle of biuret test is what? The cupric ions which is present in the biuret solution, okay, so this cupric ion in the alkaline medium will react with the protein, okay, and it will form a bond with the amino group to form the violet color complex. Okay, so the principle is the cupric ion in alkaline medium react with the amino group of the peptide bond to form a violet color complex. Okay, so this is a test to identify proteins. In the color reaction test of amino acid, do you remember xanthoproteic test? Xanthoproteic test, it is used to identify which amino acid? Any idea? Okay, so different types of amino acids can be identified with different type of tests. Okay, remember xanthoproteic test is used to identify tryptophan. So whenever there is any tryptophan in a protein, it is identified by xanthoproteic test. Then nitroprusside test is used to identify the cysteine molecule. Clear? What about ninhydrin test? Ninhydrin test is used to identify the alpha amino group. Clear? So when you do a ninhydrin test, there is one amino acid which does not give a purple color Ruhimann's complex. It gives a yellow color. Can you name that amino acid? Anup, bolo. Any amino acid?
No, ma'am.
Proline is having an amino group or an imino group? NH2 group or NH group?
Immuno group.
Yeah, imino group.
Immunogroup.
Okay, for ninhydrin test, you need an amino group. But proline is having an imino group, that is NH group. That's why it will not give a purple color complex, which is known as Ruhimann's purple in ninhydrin test. Okay, it gives a yellow color complex. So remember, these are the questions which will be asked in the viva. The small things. Okay, so proline, it will give yellow color compound on ninhydrin test. Other amino acids will give a purple color complex. Then those amino acid which is having an extra amino group like asparagine and glutamine, they will give a brown color compound. Clear? So that's all for today. We'll continue in the next class. Okay, students, thank you so much.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Okay, welcome.